Okay, so a very good morning to all of you. So my name is Edmar Banerjee. I'm working as a PhD scholar in India lab. So today I will just teach you the basics of how can we analyze and interpret in terms of statistics any kind of data that we acquire from experiments. So at first, we all know that what is statistics. It basically it's a branch of science which deals with the analysis and interpretation of data. And why science is required? Because suppose you're like conducting an experiment. Because like when you go for an MTech project, you will perform an experiment. If you want to scientifically validate your data, if you want to understand whether data is correct or not, or if your data is really scientifically correct or not, in order to prove, in order to understand that particular part, we need to hire statistics for that. So already in your like lower classes, you have already learned like pie charts, bar graphs. So all these are part of statistics. Okay, and the most important thing is data summarization. So in a like when I was small, I used to read about like raw data, then group data, right? So what we do is whenever we collect the data, at first we summarize it or we group it into frequency distribution tables, right? As we did like the value tables and all. So at first is what we need to summarize or we need to shorten our data so we can categorize our data in a like in a good way, and also to predict that what kind of data we are getting, whether it's correct or not, whether the experiment that we are having has any significance or not. All this can be done by using statistical analysis. And basically, we have two types of methods. One is called a descriptive method, where we basically try to categorize the data and arrange the data. That is called the descriptive methods. But when we want to predict any kind of result, like suppose you are having any kind of drug, you are trying to prove that this drug works in a particular like cancer treatment or something like that. In order to statistically validate this part, we need to have the inferential method of statistics. And most important thing is, you have already studied in biostatistics about normal distribution. So basically, one of the most important distributions in continuous probability function is called normal distribution. Now, what is a normal distribution? Actually, I will just start with the basics. In probability, we have two kinds of distribution. One is called the discrete distribution, and another is called the continuous distribution. So discrete distribution means we are having discrete sets of data. There is separate sets of data. What we do, we use the summation like parameter to find out the probability distribution. But when we are dealing with a continuous distribution, that is when we are trying to find out any kind of probability within a particular range, then we use integration to particularly identify the continuous distribution. And this is called the probability density function. We have two types of function. One is called the probability mass function, and another is called the probability density function. So this basically is called the probability density function. Now, what is the beauty of this normal distribution is that it's one of the only distribution that is a symmetric distribution. It does not have any asymptote. It means that as you can see from here, it's a bell-shaped curve with no skewness. Now, as I've studied in the lower classes, skewness means if the data is deviated from the mean. We know that sample mean is very important in order to understand any kind of deviation, right? So if your bell shape is like this, it means that the data is normally distributed without any kind of skewness. But suppose if the data is positively skewed, then your tail will be in the right hand side. That means right tail or left tail. Okay, right tail or left tail. And most importantly, based on this normal distribution, we try to classify any kind of data, whether it is normal distributed or non-normal distributed. And the most important thing is that in our research, in whatever research we conduct, we actually have two kinds of hypotheses. One is called the null hypothesis, and another is called the alternate hypothesis. I'll give an example, simple example. Suppose you are thinking of a drug X. That drug X, suppose you are assuming that it works on your cancer cells. So that is a null hypothesis. Okay. Alternate hypothesis means it's just the opposite. It doesn't work. So how will you test that particular thing? You have to test those test the hypothesis using these categorical tests. Now I will explain this briefly about this parametric and non-parametric. Parametric test means whenever your data is normally distributed. Suppose your data is given. Suppose you have a data. You have seen that it fits within that particular probability density function. Then only you will understand that yes, you have to apply the parametric test. Now, what are these tests actually? These tests are nothing but these are statistical tools in order to understand whether your data is actually significant or not. Like suppose you are trying to find out a particular drug or a particular molecule that works on different, like a particular situation. But how will you prove mathematically? You are only showing that yes, it's active. But in order to prove the scientific community, you need to highlight in terms of mathematical tools that your data is actually correct or not. Now, parametric test, it depends on the assumption that your data is normal distributed, and we use these kinds of like t-test, then a pair test, two group test, and others. But whenever we are having any kind of biological data, now that is the beauty of biological science, that whenever we are working with biology or any kind of data that is deviating from the normal distribution, then we use non-parametric test. So non-parametric test has the assumption that the data is non-normally distributed. In that case, we use these kinds of tests, like Wilcoxon test. So I will show you some of the things one by one. That how can we, like whatever the data we are getting, first we have, so we'll follow three steps. The first is we will test whether the data is normal or not. Number two, the second step is whether your data after your testing, suppose your data is normal distributed. So if your data is normal distributed, we will go for the parametric testing. But if your data is non-normal distributed, we will go for the non-parametric testing. Okay. So I'll show you in the software which is called IBM SPSS, by which we can understand. I'll take an arbitrary random data by which you can understand whether any kind of data we are taking is statistically validated or not. So I'll go step by step, one by one. You can write down the in a copy. How will I be able to do? Okay. Okay. This is a normal data. I've already tested, and so I'm showing it to you. So what will you do? First, you go to SPSS. This is a SPSS interface. Now, this is basically a tablet interface where you have a data view. So first, what we do? So there's a problem in SPSS. Basically, you can assign. Let's suppose these are some of the sample data. What happens if you write something alphabets? Then SPSS will not take the data. So you have to write it numerically. Suppose sample A, B, C, D. 4 samples are there. So if suppose it's an experiment. So what we'll do? We'll copy this data and simply we'll put it in the SPSS software. Okay, copy. Now what we will do? We will simply select those cells. Now remember this. This is called the factor variable. This is called the independent variable, and this is a dependent variable. So whatever normal test we will do on this dependent variable, not on the independent one, because the dependent variable will decide whether data is normal distributed or not. Okay. So what we will do is we will select those two data. We will go to analyze. Then we will go through descriptive statistics and we'll go to frequencies. Okay. Now in the frequencies, here the variable will be our variable 2. Why? Because this is the data by which we are concerned. This is the dependent variable. So we'll go to charts. We'll go to histograms. Show normal curve on histogram. Continue and OK. Okay. So we'll be getting the graph. What we are doing actually here, we are actually trying to understand initial. This is the initial test whether our data is normally distributed or not. Okay. See, if you can see from this figure, there's somewhat kind of deviation. But it's not completely bell-shaped curve. There is a skewness in this line, right? A skewness is there. So a tail is coming here. Maybe. Now this is not a confirmatory test. This is the initial analysis. Just we have to observe whether it's normal or not. Now we will go for the second round of analysis. That is, you have to understand from the skewness and kurtosis whether it is normal distributed. But these are not confirmatory tests. These are just like preliminary analysis. So what we will do? Again we will go to that part here. Okay. We will simply select this two columns. We will go to analyze, descriptive statistics. Then we will go to descriptives. Okay. Here descriptives. And now we will select the variable. We will go to options. Okay. We will select kurtosis and skewness. Continue and tell it now. This has come. Now, so what is the meaning of this kurtosis and skewness? In kurtosis and skewness, what happens is that skewness means if your graph is skewed, like deviated from the mean, and kurtosis means how much peak it is having, like how much width of the peak is having. Okay. In case of normal distribution, the skewness should lie between minus zero point 0.5 to zero point 0.5. Okay. And in for kurtosis, the value should be equal to 3. Now here you see this is 0.902. So skewness is almost normal. But if you see the kurtosis, it's non-normal. So there may be a chance that the data may be non-normal in nature. But this is just a preliminary analysis. We will not follow this and only blindly paste in our research and entry. Okay. Now what we will do, in order to confirm whether a data is really non-normal or not, we will use test called normality test. Okay. Normality tests basically are having of two types. One is called the Kolmogorov test. I will show you. One is called the Kolmogorov test and another is called the Shapiro-Wilk test. So two tests are there. There are lots of tests are there. Primarily we use two tests to understand whether the data is normal or not. So now we will go for the normality testing. One is called the Kolmogorov test. Kolmogorov. Kolmogorov test. You just see it in the internet. Just write it in some. Okay, sir. Okay, one minute. Okay, just I'll add it here itself. It's better for you. 1 minute. Increase the size. Yes. Just check the spelling once. Sometimes I may be wrong with the spelling. Just check once. Okay. So now I will show you how can we use the normality test in order to understand whether data is normal distributed or not. How will you do it? So first we will select the cells. This will be a little bit tricky. So you have to be very careful. So we'll go to descriptive statistics. Okay. And then we'll go to explore. Now, in this explore, two things are very important. Your variable 2 is your dependent. So this will go here. Variable 1 is a factor. And this is independent. So this will go here. This should be very carefully selected. Okay. So variable 2 is selected in the dependent list. Variable 1 in the factor list. Good. Now we will go to the plots. So we will select plots. Now we will deselect this part. Now we will go for the normality plots with the tests. Okay. Simple. So we will continue and then we'll go for OK. Now it will come. It's taking some time. Sorry. So it is basically having lots of parameters. If you see very carefully, these are very important. So, so basically, if this is called the significance value, this is called the p value, level of significance. So here in the normality test, it's very important to understand if your data is having a level of significance greater than 0.05, then it is having normal distribution. That is, what is actually happening? In the null hypothesis, we are assuming that the data is normal distributed. And in the alternate hypothesis, we are assuming that the data is not normal distributed. Whenever your significance level, this is called the small p value, is greater than 0.05, then we will understand and will clearly comprehend that this data is normally distributed. But if your significance level value is less than 0.05, it is non-normal distributed. Okay? So this is a confirmatory test by which we can understand whether it is normal distributed or non-normal distributed. Now, I have already told you, if the data is normal distributed, then what test we will use? Parametric or non-parametric? Parametric. So, in the parametric test, I will show you how to conduct ANOVA, that is analysis of variance, followed by post hoc to p test. Okay, post hoc to p test. I'll just give you a brief note here. You can write it down, or you can just listen and just two lines. We know that every data has a standard deviation, which means that how much the data is deviated from the mean, right? Now the thing is that The square of the standard deviation is called the variance. Now, variance can be directly correlated with the variability for data. It's very important to understand. If your variance is high, then the variability of the data will be high. So first what happens, suppose you have 5 samples or 4 samples, let's say. What happens is, this analysis of variance helps us to initially understand whether your data is significant or not. Suppose you are having 4 concentrations of drug. Let's say X drug, 0.1, 0.2, 0.3, 0.4%. You want to understand whether your drug that increases the concentration is having a significant effect on cancer cells or not. In order to initially determine that part, we need to do the ANOVA. That is the analysis of variance test. This is called the one-way ANOVA. Okay, one-way ANOVA. After in the one-way ANOVA, we suppose if something comes, then you will understand that yes, it's okay. Like significance is there, but it will not specify within which group, like within which data your significance is there. Suppose A, B, C, D, 4 samples are there. Suppose A and B are statistically similar, but A and C are significant. There is a significant difference between that. How will you understand? By using post hoc Tukey test or any kind of post hoc test. So in statistics, post hoc test means whenever we want to analyze the data, there is a compared means between the individual groups of samples. In that case, we will use the post hoc test. Any kind of post hoc test. I will show you in the non-normal part how can we use the post hoc test to again analyze our significance. Okay. Let's start. So what is the cutoff for same cutoff. Same cutoff. Everything, all basically this is important. This test is must see. Remember this, these two tests are important. Most important this one. It has been seen in different research papers also. The Shapiro's test is much more confirmatory as compared to Kolmogorov test. But there is no. There is some error. I will just check it out. But normally what happens in case of this one? Both of them come. I just tested. I just see once in the lab. Normally what happens? These both of them come. Sometimes it may not come also, depending on your data. But normally this test is also confirmatory. Shapiro's test. So now we will go for the, like, assuming this is a normal distribution, we will go for the testing of ANOVA and Tukey test. So how we will do? Just close this window. Okay. So this data is a normal distribution. So what we will do? We will simply select this. Okay. We will go to analyze, compare means, then we will go to one-way ANOVA. Now, dependent means what? Variable 2, right? Variable 2 will go. Good. And variable 1 means a factor. Now we will go to the post hoc. Okay. In the post hoc, we will select Tukey. Now this is an important part, significance level. 0.05. It means this significance level represents 95% confidence interval. Your total normal, like a range or normal distribution is 1. Total probability is 1. Always remember this. So 1 minus 0.05 is 0.95. There is 95% confidence interval. That is the standard mu plus minus 2 sigma. That is the standard range which we take for any kind of data analysis that we choose for the normal distribution. So we will continue. Okay. Now we will click on OK. Now see, very careful to understand. See, this is insignificant. Why? Because this is having the same group. So therefore, what we generally what papers do, either they put by a star or by a letter we use this A, non-significant, which means there will be no significant effect among the groups of the samples. Okay, understood what I'm saying? As it is coming the same group, because what is happening? The means of this particular samples are being compared. So if they are lying in only one group, it means the data is having no significance at all. That is, we can say similarly valued or statistically similar. Okay, clear what I'm saying? I'll show again another part where I will prove you there is also another type of data using this particular technique where you can see the significance. Okay, I'll just take another data. This one. This basically we have done the ANOVA and the post hoc test. What is happening here? We are comparing the individual groups of means with the populations or the samples. Now you have to, this is software only give you this data. You have to interpret from your mind, like how the data is generated. you see in a single group, in a single column is happening, right? So in single column if it happens, it means that there is no significance around the data. There is no effect. Now. If there have been effect, there have been like change in the means. हाँ यहाँ पे when you are having see variable 1 0 3 1 2 4 actually it goes as lowest to highest ये जीरो का शायद लोएस्ट वैल्यू होगा फिर थ्री का उसके बाद उनका नेक्स्ट मीन है फिर वन का उसके ऊपर का मीन है so in that way it has arranged the values right now you see over there subset for alpha equal to 0.05 okay now if there is only one column that means all the samples has been grouped together so whatever the variation in the means you are seeing that you are seeing by chance okay because there are many factors when you work with biological samples or say you have collected ECG okay so pre and post analysis you are doing if you are given a stimulus and before stimulus you have taken the reading after stimulus you have taken the reading so how do you say that yeah these two stimulus either they are in the significantly different or not they may be your drug or coffee or whatever you stimulant you're giving, it might not have any considerable effect. But you can see that there are certain changes in the mean. So if there are certain changes in the mean, that does not mean that it is due to the effect of the stimulus. So that observation you may have received just by chance. Are you getting my point? Okay. So he will show you the next one. Next one, sir. So this is the first point, but the long certificate. Just 1 minute, I'll just copy another data, normal data only. The same procedure. We'll just follow the same procedure. It's not a big problem. Okay. Everything is done, so it's simple. Now you see the comparison. Okay. So what is happening? What we do? Let's suppose this is the first two. So we did. So denote in our mind as A, B, C, and D. Okay. Suppose this is 10. So this is basically arbitrary sample. Let's say the sample is 1. 1, 2, 3, 4, and 5. So the 1 is in A, 2 is in B, like suppose 3 is in C, and 4 is in D, 5 is in D also. Which means that these three samples they are statistically significant. Or even in others also. The sample 10 over here it is having the lowest mean. Okay. So जो आपका lowest होगा whatever would be in the top, so it has to be given a value of A. Okay. Then one is in the column if you see one is 1 and 2 they are different. Two में in two group what you are having sample number 5. So 2 will assume the value of B. Okay. Then in 3 third column you are having sample number 3. It will be assuming the value of C. In the fourth column, we are having 0 and 1. So over there, you will be having the value of D. Both the thing will be having the value of D. So that so when you are plotting a bar plot with the standard deviation, over there you have to write these values A, B, C, D in this way. So from there only you will be able to see that which groups are together the variation in the mean, whatever you are saying, it is by chance and which are statistically different. Okay, so this is basically all about normal data. So now I will show non-normal data. So keep in mind for normal data, you do the shadow test. You find out whether your data is normal or non-normal. If it is normal, you go for ANOVA followed by touch test. Okay. Post-hoc tests are basically once you do one analysis, after that if you do another statistical analysis, then it is called post-hoc. So ANOVA followed by Tucky test or post-hoc Tucky test, whatever you want to say. So similarly, in non-normal distribution, there is another set of what do you call statistical measures which is being done. So we will demonstrate to you that. Okay, so basically, in case of non-normal data, there are two types of tests. We can write it down. One is called the Kruskal-Wallis test. I'll just write it here in the Excel file so you can follow it. Just 1 minute. So basically, we follow the Kruskal-Wallis test, and then the same thing, the post-advance test. So here your ANOVA and Tukey will not work because it's a non-normal data. So that means you have to understand, you have to use non-parametric measures, non-parametric variables or non-parametric tests, whatever we have discussed in the earlier slide. So now I will show you how to do. So this is the data. What now? See how will you understand non-normal? Another electric calculation. If you see the data is carefully, there are outliers. The more number of outliers the data will have, the more deviation the data will have from the mean. This is very important to understand. Outlier means what? Like it's not coming in a range. It's like 120, someone is 11, 10. That means the data is dispersed very in a long range. So if your data is dispersed in a very large range, it means that there is a high chance, a high probability your data will be non-normal. Now, you have to obviously confirm by using circular Wills test. But you can at least see from here that there's a 120 and 130 and another is very different. It's huge variation, huge variability is present. Okay. Now what we will do, we will just at the same thing, we'll copy this, we'll go to analyze. Here we will not do the compare means. Here instead we will go for the non-parametric tests. Okay, non-parametric tests, and then we will go to the independent samples. Okay, independent samples. Now here what we will do, first we have to customize the analysis. This is a little bit tedious, we have to go step by step. First is customize analysis, then we'll go to fields. In fields what we'll do, variable 1, which is very important. This group means a factor, that is the independent variable, and test field means a dependent variable. So you know that variable 1 is your this one in group, variable 2 in here in test field. So simply we will select it out. Okay, and variable 2 here, done. And then we will go to settings. Settings, what we will do, customize tests. We will simply select this one, Kruskal-Wallis one-way ANOVA. This is also one-way ANOVA. This is also one-way ANOVA for non-parametric testing, and we will do all pair. Why? Because it will compare between all types of pairs of samples to understand whether the groups of the samples that we are having, the groups of means, having significance. Okay. So now we have done. Now we will done. See, this is your basically your path. A table will come like this. That is not written on hypothesis. Now what we will do, we have to click on this table. See, this is your data. This is your data. Now what we will do, we will go to view. This is a view option is here. So what we will do is, we will simply go to pairwise comparison. Now you see, this is your significance level. Now you have already studied if the P is greater than 0.05, what? Not normal on normal. Normal less than is normal. So we will compare points. So what is the benefit here? It is directly giving you the comparison. It is 3 and 0, then 3 and 2, 3 and 4. Not like the previous we have in that table, we have to understand and comprehend like which one was A, B, C. Here what we are having, the software is directly giving you the data. You just need to understand whether it is significant or not significant. From here only you will be able to understand. That is the beauty of non-parametric testing. So in this way, we will be able to understand whether your data is like normal or not normal. We have already understood, and then we can calculate the statistical significance of it. And why it is required? Because suppose you are writing in discussion in any kind of experiment, you have to prove statistically, right? You are Like suppose you're generating a data, how will you prove mathematically that it's correct or not, what is significant effect or not? So you have to use these techniques in order to validate your data to the scientific community. So any like questions you have, like any kind of questions you can ask, like any kind of queries or questions in basics of statistics or anything, you can ask. No problem.
No, whatever field you choose, whether you go for biomaterials, whether you want to go for this image processing, whatever field you want to choose, this analysis will be waiting for lifelong. This is the basis of your research. Tomorrow I will just cover it in MBA, so I thought, okay, let him give the explanation because I will just attend one class. So if you see the demonstration, it will be fed into your mind in a proper way. Any question? ओवरऑल इसको रिजेक्ट कर दिया ओवरऑल
हाँ इसका मतलब क्या है यहाँ पे अगर अगर हम रिजेक्ट करेंगे इसका मतलब है कि नॉर्मल एक्सेप्ट होगा। हाँ यहाँ पे उल्टा होगा जब नॉन पैरामीट्रिक करेंगे ना तब हम नॉन हाइपोथेसिस को उल्टा लेंगे इसीलिए क्या हो रहा है देखो नॉर्मली व्हाट विल हैपन p is greater than 0.05 means what we are rejecting the hypothesis which means it's the thing
लेकिन इंडिविजुअल अगर हम देखें थर्ड वाले उसमें देखें 0.014 है
हाँ मतलब यहाँ पे अगर हम ये भी ना भी देखें तो this one is most important level of significance if p is greater than 0.05 normal is like significant अच्छा here one thing here one
most of that is the most
so here one thing I'll just explain you in this na normal and non normal are not required here what we are having level of significance okay remember this if your p value this is small p value that's called level of significance just write it down if p is less than 0.05 if small p value is less than 0.05 then the data are statistically significant. Then the data are statistically significant. But if the p value is greater than 0.05, it means that the data are statistically similar or similarly valuable. Why? I'll just give you a brief hint. There's not chalk anything. Okay, I'll just explain you. Have you seen the normal distribution graph? Typically, I just explain you. Otherwise, you'll not be able to understand. If you have seen the bell-shaped graph, right? You will see very carefully. If you like, you go to internet, you will see there's a like bell-shaped graph where there's a 95% confidence interval. Okay, mu plus minus 2 sigma. Okay, what happens? Your normal distribution lies within this range. But unfortunately, if your data deviates or goes beyond this range, 95% confidence interval, then it becomes non-significant. Why? Because it goes to the tail region. That means it's much more deviated from the mean, right? That means what the significance decreases. So as the data converges towards the mean, there is mu. Because we have two parameters, mu and sigma square. Mu is your mean and variance is sigma square. Okay. So if the data converges towards the mean, it means that there will be more probability that the data is significantly major. Clear what I am saying? That's why we are always considering greater than 0.05 to be this one. That is the main part, major part. 95% confidence interval is the most important part. That's all. So you can study more like on internet on these things. More things are there. It is just a glimpse of things. Lots of things we do in our lab and lots of things we have to analyze. This is just a glimpse of whatever we are doing. It's very basics and things. So any like any kind of questions you have? Any more? Any questions? Any kind of queries?
नॉन पैरामीट्रिक टेस्ट में कौन सा वाला बेस्ट है उससे पहले
हाँ और भी है विल्कक्सन टेस्ट भी है बहुत सा टेस्ट लाइक देयर इज नो लिमिट नाउ दे हैव टू कंपेयर द मेन थिंग इज दैट टू कंपेयर व्हिच वन इज हैविंग बेटर रिजल्ट्स
ऐसा नहीं होता कि एक टेस्ट दिखा रहा है डिफरेंस रिजेक्ट कर
नहीं ऐसा नहीं नहीं ऐसा नहीं क्योंकि भी नहीं होता है हाँ कुछ कुछ में होता है कब होता है ये भी बहुत इंपॉर्टेंट क्वेश्चन है इट्स गुड क्वेश्चन sometimes what happens when your post hoc tests are incorrectly done Because post-factum, what it does not, it reduces the type 1 error. There are two types of errors. One is called type 1 error and type 2 error. Type 1 error क्या है? False positive जो होता है ना, like false positive भी है. Like suppose if your data is not significant but it's showing it's significant, it's false positive. In order to reduce that part, we use post-hoc tests. Sometimes, if you statistical software anything, you have made any error in your programming or whatever, you can also do a programming also. Suppose it's done something error, it will show you false positive. At that time, your post-hoc test may not work. So you have to design it in such a way that your post-hoc test really work in that way. So statistically, you can validate your data.
We are not going into that part because it's not within that scope of focus. So anyhow, yeah. So there are various kind of corrections like one-family correction, one-family correction. Whenever you are doing multiple tests, there also you are including some errors. Okay, so we are not going into that. So over there, then again, one-family equation, correction factor, and all this. वो सब फिर आ जाएगा. So for our day-to-day practical classes, main thing is that you have to first check whether the data is normally distributed or not. If it is normally distributed, ANOVA followed by Turkey test is the most commonly used method for any of the journals. Okay, for journal publication. And if it is a non-normal data is distributed, then you either Bushel-Wallace test or Wilkinson's sign rank test, followed by Dunn's test. These are the methods to look into the signature. Because we have seen people, even when they are presenting their this MTech result group, there is a change in the name. My there is a they don't do the statistical test and they will directly say after doing this treatment it has increased. But until and unless you do the thing properly, you do the statistical analysis, it has no value. Increasing mean has no value. Whatever the increase you are seeing, that increase may be due to you have got it by chance. Suppose you have done some mistake. So due to some mistake, the mean has in one of the sample it has come slightly high, and because of that, the total mean has come. So until and unless you do the statistical analysis and from there you find out the pairwise comparison, you cannot tell whether there is a change in the property of your material or change in the features, feature values. Okay. So that is the एक टीचर का ये होता है जितना हम लोग जानते हैं उतना आप भी जानते हैं लेकिन तब जाके आप याद रखोगे